Welcome. My name is Jinish. I am from Miramaya University of Applied Sciences. In this session, we will be discussing introduction aspects to proteins. So, in this session, we will study the functions of proteins and structure of proteins. Proteins in living system. Proteins are the most abundant organic molecules of our living system. They occur in every part of the cell and constitute about 50% of cellular dry weight. Proteins form the fundamental basis of structure and function of life. The word protein comes from the Greek word proteos, which means the holding the first place. Functions of proteins. We can classify proteins function into two: static functions and dynamic functions. First, we will discuss static functions. Certain proteins perform brick and mortar roles and are primarily responsible for structure and strength of the body. So certain proteins, they mainly give the structure and strength of the body. Example, collagen and elastin, they are found in bone matrix, vascular system, and other organs, and alpha keratin present in epidermal tissues. So all these proteins actually serve the static functions to the living body. We will discuss now dynamic function. Proteins performing dynamic functions are appropriately regarded as the working horses of the cell. So working horses of the cells are protein through their dynamic functions. Proteins acting as enzymes, hormones, blood coating factors, immunoglobulins, membrane receptors, storage proteins. Besides their function in genetic control, they are useful in muscle contraction and respiration, etc. Now we will see the elemental composition of protein. Proteins mainly consist of 5 elements: carbon 50 to 55%, hydrogen 6 to 7.3%, oxygen 19 to 24%, nitrogen 13 to 19%, sulfur 0 to 4%. In addition to that, proteins may also contain elements in smaller amount such as phosphorus, iron, copper, iodine, magnesium, manganese, and zinc. Proteins are polymers of amino acids. So we know that around 300 amino acids are present in the nature. Out of that, 20 standard amino acids are repeatedly found in the structure of the proteins. So the proteins in the living beings are mainly made up of 20 amino acids arranged in a particular manner. And they are the polymers of L-alpha amino acid. We know what is alpha means and what is L. We have studied it in the basics of organic chemistry. Protein synthesis in the cell is controlled by DNA, the genetic material of the body. So with the instruction of DNA, different amino acids arranged in a particular order leads to the formation of the targeted protein. After the synthesis of protein, some of the incorporated amino acids undergo modifications to form their derivatives. So amino acids join to form the polymer, which is protein. After the protein formation, some of the amino acids in that protein chain will undergo some kind of modifications and form their derivatives. Amino acids are a class of organic compounds containing two functional groups. One amino group, which is NH2, which is basic in nature, and one carboxyl group, which is acidic in nature. That means in an amino acid, minimum one amino group and one carboxylic acid group should be present. Now we will study the general structure of amino acids. Alpha amino acids, if both the carboxyl and amino groups are attached to the same carbon atom. Why it is alpha amino acid? If you see here, this carbon atom, to this carbon atom, both the carboxyl functional group and amine functional group, they are attached. So that's why we call it as alpha amino acids. Optical isomers of amino acids. If a carbon atom is attached to 4 different groups, it is asymmetric and therefore exhibit optical isomerism. The amino acid except glycine possesses 4 different groups. For example, here you see R, H, NH2, COH2. So for example, glycine, this R is H. So in that case, this carbon contains 2 groups similar. In that case, glycine cannot form optical isomerism. Any other group, for example, R is CH3. At that time, all the groups attached to the alpha carbon atoms are different. So definitely that compound will show optical isomerism. So except glycine, all amino acids show optical isomerism because they possess 4 different distinct groups. Thus, all the amino acids except glycine, where R equal to H, have optical isomers. D and L form of amino acids. So here you see, here it's a representation of glyceraldehyde. Now you see the position of H and OH for D-glyceraldehyde. Now you see the position of OH and H in L-glyceraldehyde. Now you know why it is D, why it is L. It's the position of the H and OH. Similarly, D amino acid, see the position of H and NH2 in the amino acid structure, and here you see the position of H and NH2. If you see the position, then you will be able to say that which one is D amino acid and which one is L amino acids. So the D and L, that naming is actually adopted from the glyceraldehyde structure. Now you see, this is L amino acids. NH2 position is here. This is D amino acids, NH2 position is here. And the proteins are made up of L alpha amino acids. So its configuration should be like this L configuration. Now we will classify amino acids. Amino acids can be classified in terms of their structure, polarity, nutrition, and metabolic field. For example, classification of amino acids. Amino acids classification based on the structure. We can classify based on polarity. We can classify based on their nutritional classification, and we can classify based on their metabolic fate. Metabolic means how it will metabolize. First, we will study amino acid classification based on the structure. The 20 amino acids found in protein are divided into 7 distinct groups. So based on the structure, we can classify the 20 standard amino acids into 7 distinct groups. For example, the first one, amino acids with the aliphatic side chain. So the R group, if you see that R group, in all these amino acids, in glycine, alanine, valine, leucine, isoleucine, the side chains are aliphatic groups. That's why all these amino acids are coming under the category amino acids with aliphatic side chain. First type. Second type, amino acids contain hydroxyl group. You see here in the structure, OH is present for serine, threonine, and tyrosine. So if you carefully see the structure of these amino acids, we will be able to see their side chain contains OH functional group. So this is the second group. Third group, sulfur containing amino acids group. For example, cysteine, and methionine. Side group contains at least one sulfur atom. Cysteine, methionine. All contains sulfur atoms. For example, cysteine contains sulfhydryl group which contains sulfur. Cysteine contains disulfide group which contains 2 sulfur. Then thioether, where sulfur is present in methionine. In all these cases, sulfur is coming in the side chain. So we call these amino acids as sulfur containing amino acids. Next is acidic amino acids and their amides. So definitely you see their side group will be either acidic group or the amide of that acidic group. See, either acidic group or its amide group or their amide group. Examples: aspartic acid, asparagine, glutamic acid and glutamine. If you carefully see here, an acidic group, aspartic acid, here you see NH2CO, so it's amide. Glutamic acid, acidic group, and its amide group. You can see NH2CO, that bond we can actually see the CONH2 group. So CONH2 group is amide. So glutamic acid, if its amide group is forming, then it is glutamine. In aspartic acid structure you see is acid. Instead of that, if it is amide, then it is asparagine. Fifth group, basic amino acid group. Basic amino acid group means the side functional group will contain a basic group. Here the basic group is amino group. Here the side group contains basic. This group is guanidino. Here also a basic group. As a side group, it is a imidazole group. So basic amino acids, lysine, arginine and histidine, they come under basic amino acids group. So we studied now, based on the structure, 5 groups. The 6th group now is aromatic amino acid group. As a side group, an aromatic group, aromatic group, aromatic group. In phenylalanine, benzene or phenyl is the aromatic part. In tyrosine, it is phenol group. In tryptophan, that is indole group. So these amino acids contain aromatic groups in their structure. Next is last one, amino acid. It's proline. So in proline, we will be able to see that there is a pyrrolidine group. That is the imine group. Pyrrolidine is a special group present in the structure. So this comes under the last group, which is amino acid contains, or amino acid or amino acid group containing amino acid. So these are the chemical structure of different amino acids. So neutral amino acids contains only one carboxylic acid and one NH2 group. Basic amino acids contains one COH group and two NH2 groups. Example: lysine, histidine, arginine. Example for neutral amino acid, glycine is an example. Acidic amino acid group means, example, aspartic acid, glutamic acid, or asparagine and glutamine. They are either acid or its amide derivatives. Those groups will be present in the amino acid structure. Amino acids, proline. There can be aliphatic amino acids and aromatic amino acids under neutral amino acids. For example, in aliphatic amino acids, simple amino acids, glycine and alanine. Branched chain, if the side group is containing branch, then they are valine, leucine, isoleucine. If hydroxy groups contains, then it is serine, threonine. If sulfur is present, they are cysteine, methionine. Then aromatic amino acids group, we classified them: phenylalanine, tyrosine, tryptophan. All these comes under the aromatic group contains amino acids. So first classification is over. Second classification of amino acids based on polarity. Amino acids are classified into 4 groups based on their polarity. Polarity is very important for protein structure. Non-polar amino acids means hydrophobic or water-hating amino acids. there no charge on the R group. If you go back to the basic structure of amino acids, the R group. If that R group doesn't contain any charge, then we will call that amino acid as non-polar amino acids. They will be majorly hydrophobic or water-hating behavior. Examples: alanine, leucine, isoleucine, valine, methionine, phenylalanine, tryptophan, and proline. Polar amino acids with no charge on R group. Here, the R group will not be no charge, but they will be polar. For example, hydroxyl, sulfhydryl, amide. They can participate in hydrogen bonding of the protein structure. Example: glycine, serine, threonine, cysteine, glutamine, asparagine, and tyrosine. Their R group doesn't have any charge, but they are polar in nature. Because of their polarity, they will be able to participate in hydrogen bonding of protein structure. Protein structure we will discuss later. Polar amino acids with a positive R group. So here, the side group will be polar, the R group will be polar. Not only they are polar, that R group will be having positive charge. For example, lysine, arginine, and histidine. Polar amino acid with a negative R group. So the R group will be polar as well as will be having a negative charge. They are the dicarboxylic mono amino acids, for example, aspartic acid and glutamic acid. So based on the polarity, we can classify proteins, amino acids into non-polar amino acids where the R group will be having no charge as well as non-polar. Next is polar amino acids with no charge on the R group. On the R group there will not be any charge, but that will be polar, and it can be, and those groups can take part in hydrogen bonding in the protein structure. Then polar amino acids with a positive R group. The R group will be having positive charge. Along with that, it will be polar in nature. Then last one, polar amino acids with a negative R group. So R group will be polar in nature as well as it will be having negative charge also. So classifications are given and some examples for non-polar R group: alanine, valine, leucine, isoleucine, proline, phenylalanine, tryptophan, and methionine. See the structure. There is no chance of the polar groups as are in these structures. Next is polar R group without any charge. The glycine group, serine, threonine, these are the R groups in the amino acid structure. Tyrosine, asparagine, glutamine, all doesn't have any charge. Even though they don't have any charge, but they can undergo hydrogen bonding in the polymers in the protein structure because of their polarity. Next is the negatively charged R group. See, in our aspartic acid, the R group contains a negative charge. In glutamic acid, R group contains a negative charge. Similarly, positively charged R groups containing amino acid, for example, lysine you see a positively charged R group as the side group. Here also you can see a positively charged R group, So lysine, arginine, histidine, they contains the R groups with positive charges. Third classification is based on nutritional requirements. Based on the nutritional requirements, amino acids are grouped into two classes: essential and non-essential. Essential or indispensable amino acids. The amino acids which cannot be synthesized by the body need to be supplied through the diet are called essential amino acids. So some of the amino acids that our body will not be synthesizing, therefore we require those amino acids through the food intake, through the diet that we follow. Those amino acids are known as essential amino acids or indispensable amino acids. These amino acids examples are arginine, valine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, and tryptophan. The two amino acids, namely arginine and histidine, can be synthesized by adults and not by growing children. another two amino acids, arginine and histidine. This can be synthesized by adults but not by growing children. Hence, these are considered as semi-essential amino acids. Thus, 8 amino acids are absolutely essential while 2 are semi-essential. Non-essential or dispensable amino acids. These amino acids will be synthesized in our body. The body can synthesize about 10 amino acids to meet the biological needs. Example, glycine, alanine, serine, cysteine, aspartate, asparagine, glutamate, glutamine, tyrosine, and proline. These amino acids will be synthesized in our body. So it is not necessary that these amino acids should be there in our diet. So these are the classification of the essential amino acids and semi-essential amino acids. Fourth classification is amino acid based classification based on their metabolic phase. The carbon skeleton of the amino acids can serve as the precursor for the synthesis of glucose, glycogenic, or fat, ketogenic, or both. So amino acids carbon skeleton can serve as the precursor for the synthesis of glucose, that is glycogenic, or fat, ketogenic, or both. Glycogenic amino acids. These amino acids can serve as precursor for the formation of glucose or glycogen. For example, alanine, aspartate, glycine, and methionine. So these amino acids serve as the starting material, as a precursor for the formation of glucose or glycogen. Ketogenic amino acids means fat can be synthesized from these amino acids. The two amino acids, leucine and lysine, are exclusively ketogenic. So these amino acids, you can actually convert it into fat. So fat can be synthesized from these amino acids. Now glycogenic and ketogenic amino acid. 4 amino acids, isoleucine, phenylalanine, tryptophan, tyrosine. They are precursors, starting material for both glucose as well as fat synthesis. So this is the last classification of amino acids, and that is based on their metabolic fate. Metabolic means how these amino acids will be undergoing metabolism and how it will be utilized by our body. Now we will study about the properties of amino acids. In this we will study the properties of amino acid, and we will study the statement, or we will justify the statement amino acids are amphiolites, and we will also define what is the isoelectric pH of an amino acid. So properties: most of the amino acids are usually soluble in water and insoluble in organic solvents. Melting point, are they generally melt at high temperature, mostly above 200 degrees Celsius. Most of the amino acids are sweet. Glycine, alanine, valine, all are sweet taste. Some doesn't have any taste, that is leucine, or some amino acids will be having bitter taste, that is arginine or isoleucine. Optical properties: we know that all amino acids except glycine possess optical isomerism due to the presence of a symmetric carbon atom. Some amino acids also have a second asymmetric carbon, example isoleucine and threonine. So we already know that except glycine, all the amino acids will have optical isomerism because of the asymmetric carbon, which is the alpha carbon. And some amino acids will be having more than one asymmetric carbon, for example isoleucine and threonine. Now amino acids as amphiolites. Amino acids contains both acidic and basic groups. So they can donate a proton or accept a proton. Hence amino acids are regarded as amphiolites. Citration or dipolar ion. Same meaning. Amino acids contain both acidic and basic group. Acidic group is OH and basic group NH2. They can donate a proton or accept a proton. Hence amino acids are regarded as amphiolites. They can be in citration or dipolar ion form. We can write it in that form. The amino acids rarely exist in the neutral form with free carboxylic, that is OH, and free amino groups. If you take the structure of amino acid, if you remember the structure of the amino acids, if you see the structure, then you will be able to tell that the COH2 and NH2, they will not be like neutral form. In acidic pH, in strong acidic pH, that means below low pH. In acidic condition means lower pH. The amino acids is positively charged cation, while in strongly alkaline pH, at higher pH, it is negatively charged anion. Why? It shows those characteristics we will study later. Each amino acid has a characteristic pH. For example, leucine, it is pH equal to 6, at which it carries both positive and negative charges and exists as Sutter ion. So what is Sutter ion, that we will discuss later. Now you see how the Sutter ion forms. So this is the general structure of amino acid RCNH2COOH, and we know that they cannot stand like in neutral groups. What will happen? This H will go to here, form NH3 plus and COO minus. So in this form, amino acids exist. In this form, amino acids exist. Uncharged structure, COOH, NH2. But I told you there will be an exchange of H plus ion, and you can represent it in the form of Sutter ion or dipolar ion. What is isoelectric point, that we will discuss now? An amino acid structure is written here. I told you at a particular pH it will form Sutter ion, and at that time the Sutter ion, the positive charge and negative charge will be there, and that will be same. Both positive and negative charge quantity will be same. Therefore, Sutter ion exists as an ion with no charge, because it has negative and positive charge, but the number of negatively charged group and number of positively charged groups are same, so its net charge is 0. So we can call it as the molecule is electrically neutral. But at lower pH means mainly in acidic condition. In acidic condition, the H plus in the acidic condition can go and react with the COO minus, form COOH, and the structure will be like this. Now you see the amino acid is having now net positive charge, or amino acid will be acting as a cation. At the same time, if at high pH what happens? If you see at high pH means H plus will be removed. For example, at higher pH means mainly it will be an alkaline solution. This OH minus will be there. So from NH3 plus, 1H plus will go and interact with that OH minus, forms water. So NH3 plus will become NH2 and COO minus will become as such. Now you see the net charge of this molecule is negative. So we call it as an anion. So now we know that the amino acids exist in charge based on the pH of the solution where amino acid is present. At a particular pH, the number of NH3 plus ions, NH3 plus charge or CO minus charge will be same. At that time, it will be electrically neutral, and that pH is known as isoelectric pH. It's represented as Pi. So Pi value can be calculated by taking the average pK value corresponding to the ionizable group. For instance, leucine has 2 ionizable groups, and its Pi can be calculated as follows. Leucine exists as cation. Leucine at pH below 6 and above 6, it will act as anion. But at a particular pH which is at 6, leucine exists as Sutter ion. So pH of the medium in which we have taken the amino acid determines ionic nature of the amino acids. If the pH of the medium is lower than that of the isoelectric pH, then the amino acid will be acting as a cation with positively charged. And if the medium, pH of the medium is higher than that of isoelectric pH, then the amino acid will be existing as anion with negative charge. So we know amino acids, general formula like this, but in ionizable form, we know that the NH2 will become NH3 plus and COOH minus becomes COO minus, like this, carboxyl group, amino acid group. At isoelectric point, carboxyl group and amino acid groups are same. Therefore, that amino acid will be electrically neutral. If NH3 plus amount is higher than that of COO minus or reverse, then the amino acids will be having their own charges. At lower pH, COO minus will be receiving H plus ion from that lower pH medium, form COOH, and this will become NH3 plus, which means now the amino acids are positively charged, and they are acting as cation. At higher pH, this H plus will be going to the OH minus of the medium, leaving NH2 here in the structure and COO minus. Now this portion will be neutral and COO minus will be in negatively charged. Then this amino acid, now it is in negatively charged and we will call it as anion. So these are the isoelectric pH of different amino acids. For glycine, alanine, valine, leucine and isoleucine, its value is near 6. For phenylalanine, it is 5.5. For proline, 6.3. For serine, 5.7. And for threonine, 5.6. Now titration curve for glycine. Titration curve means you have taken a glycine solution, and that glycine solution you are changing the pH. Example, you are bringing the pH of the glycine to a very low pH by adding acids such as nitric acid. Then you are titrating it against sodium hydroxide solution, and you are measuring pH of the glycine solution throughout this experiment. And we can see that when we are adding the alkali, sodium hydroxide or potassium hydroxide, into the acidified amino acid solution, for example glycine, then we can see that how the pH is changing. pH is drastically changing at a particular pH, then again drastically changing at a particular pH, again it's a constant change, then again drastic change. So you will be able to see two different drastic change in the values of pH when we are adding the alkali solution into the amino acid solution. The pH controls the charge of the glycine. Cationic below pH 2.3. So you see here the pKa value, that is negative logarithm of Ka. Ka is dissociation constant. So if you see below 2.3, that is higher pH, at higher pH amino acid will behave as cation. So at lower pH means high acidic condition, amino acid will be acting as a cation. Similarly, above 9.6, the amino acid will become alkaline in nature. At that alkaline condition, amino acid will be behaving as anion in nature. Isoelectric point is the pH at which the amino acid is evenly balanced between the two forms as dipolar citrion with a net charge of 0. Next topic is structure of proteins. We can actually see proteins have four different structural units or structural behavior. They are primary structure, secondary structure, tertiary and quaternary structure. We will be studying one by one. Primary structure of protein. Primary structure is linear sequence of amino acids forming the backbone of the protein. Proteins are also known as polypeptide because the bond formed when two amino acids join is a peptide bond. That's why a protein contains so many amino acids, so many peptide bonds. Proteins are also known as polypeptide. So each protein has a unique sequence of amino acids which is determined by the genes contained in DNA. Each protein has a unique sequence of amino acid and that sequence is determined by the genes contained in the DNA. So DNA determines in which order the amino acids joins to form the right protein. The primary structure of protein is largely responsible for its functions. The function of a protein is mainly depends on the primary structure of the protein. A vast majority of genetic diseases are due to abnormalities in the amino acid sequences of the protein, that is the changes associated with the primary structure of protein. So genetic diseases are mainly due to the misalignment or any changes in the primary structure of the protein, means the arrangement of amino acids in the right order. The amino acids composition of a protein determine its physical and chemical properties. See the primary structure of amino acids. So all these are amino acids. So many amino acids forms the preliminary or primary structure of protein. Protein primary structures are given. So 1, 2, 3, 4. Here 5 amino acids are shown, and each amino acids are bond when they are bonding. A peptide bond is being formed. That's why proteins are known as polypeptides. And one end of the protein chain will be carboxyl terminated and the other end will be amine group terminated. Peptide bond. The amino acids are held together in a protein by covalent peptide bonds or linkages. These bonds are rather strong and serve as a cementing material between the individual amino acids. We consider it as bricks. Formation of a peptide bond. When the amino groups of an amino acid combines with the carboxyl group of another amino acid, a peptide bond is formed. See, it's a citrionic form of the first amino acid. It reacts with the second amino acid, which is also in citrionic representation is given here. Now we can see that the carboxylate group will be reacting with this amino group, and a condensation reaction will happen where H2O will be removed, and between these two amino acids now a peptide bond is formed, which now formed a dipeptide. Dipeptide is having the one end carboxyl terminated, the other end amino terminated. Now when third amino acid is reacting, for example, I will write the third amino acid. What will happen? The carboxylate group of this dipeptide will react with the third amino acid's amino group. Again, water will be eliminated and there will be a CONH bond will happen between the second amino acid in the dipeptide chain and the third amino acid NH3 group. So a new CONH, then it will be a tripeptide. Similarly, if it continuing, then we will get a polymer chain primary structure. Characteristics of peptide bonds. The peptide bond is rigid and planar with a partial double bond in character. So see this peptide bond. They are very strong, rigid in planar as well as they have partial double bond in character. They generally exist in trans configuration. Both CO and NH group of the peptide, so CONH, right? Both C double bond O and NH groups of the peptide bonds are polar and are involved in hydrogen bond formation. Writing of peptide structure. Conventionally, the peptide chains are written with the free amino end, that is N-terminal residue on the left side, and the carboxyl end, that is the C-terminal residue at the right. Here we have written like that only. The amino acid sequence is read from the N-terminal end to the C-terminal end. Incidentally, the protein biosynthesis also starts from the N-terminal amino acids. Now, how we will write a shorthand to read peptide? Different way to represent a peptide chain. For example, here you see glutamate, cysteine, glycine. 3 amino acids are in this peptide chain, where the left glutamate, the open end, the first end or the starting end is N-terminal, and C-terminal is in glycine. We can easily write its representation, symbol representation. Glutamate, we have seen that it is E, cysteine is C, and glycine is G. Or we can write in this form for glutamate GLU, cysteine cis, and GLY glycine. So in this way also we can represent a peptide chain. Or in glutamyl, cysteine, glycine. So in different way we can read the peptide or represent the peptide. So using 1-letter symbol or 3-letter symbol or a peptide name. Now you see the dimensions of the peptide chain. Alpha carbon atom of one amino acid. Its neighbor amino acids alpha carbon atom. The distance between these two alpha carbon atoms is in the range of 0.36 nanometer. In the peptide bond, there is carbon double bond oxygen bond. This is the C double bond O bond in the peptide bond. The length is 0.123 nanometer. In the peptide, that is NH bond, the bond length is 0.1 nanometer. This CN bond is having a bond length of 0.132 nanometer. From the nitrogen in the peptide group to the alpha carbon atom, the length is 0.147 nanometer. The bond angle between NCC, here it is 110 degree. The bond angle between OCN is 122 degree. For CNH, 120 degree. For CNH, that is also 120 degree. So this is the dimension of the polypeptide chains. Now secondary structure of protein. The spatial arrangement of protein by twisting of the polypeptide chain is known as secondary structure. The conformation of polypeptide chain by twisting or folding is referred to as secondary structure. The amino acids are located close to each other in their sequence. 2 types of secondary structures, alpha helix and beta sheet, are mainly identified. So 2 types of secondary structure of proteins are identified. We call them as alpha helix and beta sheet. Now we will study the alpha helix structure. In alpha helix structure, we can see that the amino acid joined peptide chain or protein chain are like in helical form. Now each, what we say that role in that helical chain are connected using hydrogen bond. So we know that we have studied the CONH, the peptide bond, the C double bond O and NH, both are polar in nature. They can undergo hydrogen bonding in the polymer chain. For example, in this helix structure, there are NH group, C double bond O group, NH, COO. Similarly, in the next helical, there will be C double bond O, there will be NH. Between these two helical portions, there are C double bond O and NH. Between them, hydrogen bonding will happen. And this forms the alpha helix structure. And this hydrogen bond gives the stability to the alpha helix structure. So alpha helix structure can be represented like this, and the planar structure of the peptide bonds are shown here. Again, the hydrogen bondings are shown here. See, in one row there are C double bond O, NH, C double bond O, NH. We know that hydrogen bonding is happening in each helical, what we say, the helical circles. In beta pleated sheet, this is a second type of structure proposed by Pauling and Corey. Beta pleated sheets, or simply beta sheets, are composed of two or more segments of fully extended peptide chain. This is the first peptide chain, second peptide chain. So it's not a single chain, it's two different peptide chains. So these two peptide chains composed to form the second type of structure, which is beta pleated structure. And the representation of beta pleated structure is given here. So a beta pleated sheet structure is given here. Now the third one is tertiary structure. A protein's three-dimensional 3D structure, also referred to as its conformation or higher ordered structure. Higher ordered structure is known as HOS. It is a compact structure with hydrophobic side chains held interior, while the hydrophilic groups are on the surface of the protein molecules. This type of arrangement ensures the stability of the molecule. So in three-dimensional tertiary structure, it's also referred to as conformation or higher ordered structure. It's a compact structure with the hydrophobic side chains will be inside the 3D structure, while hydrophilic group are on the surface of the protein molecule. This type of arrangement ensures the stability of the molecule. Now, in the tertiary structure, besides the hydrogen bond, disulfide bonds, ionic interactions, hydrophobic interactions, and van der Waals force of interaction leads to the formation of tertiary structure. The term domain is used to represent the basic unit of protein structure of tertiary, that is tertiary structure and function. A polypeptide with 200 amino acids normally consists of two or more domains. So that tertiary structure, you see that this is a secondary structure. Now imagine that you are actually, entangling the entire secondary structure, then you will end up with this structure, and that structure is known as tertiary structure. So you see, in that tertiary structure, you can see that secondary structure, and that secondary structured polymer chains are actually combined or what we can say that folded to form tertiary structure. Next is quaternary structure, where secondary interactions with the 3D tertiary structures. So here in the tertiary structure, the linkages between, so here sulfur-sulfur linkages, van der Waals force of interaction or hydrophobic interactions, ionic interactions, all these are actually makes this tertiary structure stable. So we studied the structure of the protein. So we studied about primary structure of the protein, which is linear. Then we studied alpha helix and beta sheet structure, which are secondary structure. Then we studied tertiary structure and the possible bondings other than the hydrogen bonding in tertiary structure. And the final, the interaction or the secondary interaction between the tertiary structure forms the quaternary structure of the protein. Now, bonds responsible for the protein structure. For example, here you see two amino acids, disulfide linkage, hydrogen bonding, ionic interactions, hydrophobic interactions. All these interactions leads to the protein structure stability. Last topic is denaturation of protein. The phenomenon of disorganization of native protein structure is known as denaturation. Denaturation results in the loss of secondary, tertiary, and quaternary structure of the protein. This involves a change in physical, chemical, and biological properties of the protein molecule. So, if you heat a protein, or if you change the pH of the protein, or if you are adding some alkali into the protein, or if you are adding an acid, or if you heat the protein, then there is a chance of denaturation of the protein. And during denaturation, what happens? All the bonds which were present in their structure, like disulfide bonds or ionic interaction or hydrogen bonds, those bonds, some of the bonds will be broken and the structure of the protein will be lost. And if the structure of the protein is lost, then the function of proteins also will be lost. So the protein will lose the physical, chemical, and biological properties of the protein.